So we mentioned before the fact there's negative regulators of cell division, and just want to briefly talk about the second category of tumor suppressor genes. And these are the ones that inhibit cell division. So retinoblastoma is eye cancer. It was just the gene that was associated with retinoblastoma, RB, is where we get the name for the gene from. And if you are experiencing retinoblastoma, you have symptoms like ocular inflammation, squinting to improve vision, and leukocoria, which is kind of like the bright light in the back of the eye that sometimes shows up. It RB was the first tumor suppressor gene to be identified in humans by studying patients with the disease retinoblastoma. Some people have an inherited form of the disease, and others have the disease that is the result of environmental agents that have to their eyes later in life. So this kind of brings us to the two-hit model. And this two-hit model is that essentially people have two copies of their RB gene. You get one copy from your mother and one copy from your father. Individuals with the inherited form of the disease have a mutant gene from one of their parents, but that won't lead to the disease. You have to have the gene broken completely to halt regulation. And so if they get a second hit on their other chromosome and it breaks their other RB gene, that the odds of that happening are more likely to occur early in life. So individuals with the inherited RB gene, or I should say the RB allele that is associated with the disease, the broken version of the RB gene, they tend to get retinoblastoma early in life as children, because getting to the point where both chromosomes have incurred damage on that gene is likely to occur early. Now, there are individuals who get retinoblastoma later in life, but these individuals, this tends to be very rare, and it tends to happen much later in life. And that's because it takes a long time for an individual to have broken two copies that are broken in a particular cell in their eye. Requires an incredible amount of damage, UV damage, DNA damage to the cells in the eye, and you have to get one cell that has a broken copy of the gene on both chromosomes, and then that cell would divide and result in retinoblastoma. So two rare events are far less likely than a single event. So the non-inherited form of this disease occurs rarely and later. And this is the two-hit hypothesis model cancer prognosis. Cancer can either be a sporadic occurrence or it can be familial, which means you inherited a gene that was broken, an oncogene. So with individuals that have the familial inherited version, they will get a broken copy from one of their parents, but they typically won't get cancer from just that alone. You need a non-working copy completely in your genome. And so this situation can occur. This is when you do get cancer, but and it's inherited, but it does require your second chromosome to have a broken copy at some point. With the sporadic or non-inherited version of cancer, you get the manifestation of your oncogene because both copies of that gene are broken in your genome. And so you'd have to get this situation where there's two hits in the genome that landed in that gene, that broke that gene, for cancer to progress. Back to RB. So RB codes for a protein, and the RB protein inhibits the transcription factor EF2. And EF2 is what's going to activate transcription for genes that progress your cell from G1 to S phase. So the binding of your RB protein inhibits EF2 and it prevents cell division. So when both copies of your RB gene are broken, your EF2 protein is always active and you have uncontrolled cell division.